Welcome everyone to our Bioheat and Mass Transfer Lab video. In this video, you'll see our work in thermal dynamics and heat transfer related to cryobiology and hypothermic biology, with broad applications in therapeutics, regenerative medicine, and diagnostics. Areas of emphasis in the lab include cryopreservation of living systems, such as biomedical model systems like zebrafish or Drosophila embryos, cell and tissue therapies like cryopreservation of stem cells and pancreatic islets, and cryopreservation of organs for transplant and biomedical use using RF nanowarming, a method we invented in the lab. There's also work on controlled focal therapies to improve cancer immunotherapy and improved lateral flow assay diagnostics, where we're trying to make point of care diagnostics that can rival laboratory-based diagnostics to do it faster, cheaper, and better at the point of care. We actively engage with numerous faculty and groups on campus through the Institute for Engineering and Medicine, which I direct, iem.umn.edu. And we're helping to lead a new NSF engineering research center called the Advanced Technologies for the Preservation of Biological Systems, or atpbio.org. And that works on improved cryopreservation technologies to solve supply chain issues with living biomaterials. We hope you enjoy the video and invite you to visit our websites and get in touch if you're interested in any of the projects. Thank you very much.
So the main purpose for the CPAs that we use is effectively to control the formation of ice. And what I mean by that is to eliminate the formation of ice entirely. So if I take this CPA and I cool it faster than 2.5 degrees Celsius per minute, I can actually bypass ice, effectively supercooling it. And if you cool it low enough, once you cool the solution down to its glass transition, for which this CPA is about negative 123 degrees Celsius, you can actually turn the whole solution into an infinitely viscous, amorphous solid that we call a glass. So in this state, it's very stable, and we're going to show you some videos. So if the CPA was cooled incorrectly, say it was dunked into liquid nitrogen, the CPA upon cooling can actually crack, as if it were a glass.
Hi, this is Lakshya, a PhD student in BHMP lab. This is 1 liter VS55, a common CPA, which we will try to vitrify in a controlled rate freezer. See, this is partially vitrified, and then you have these spare balls in it. Further, this shows that we have the capabilities to go to a scale-up, human scale vitrification. Cryomicroscopy is a powerful tool to visualize ice crystallization using a droplet or a thin film of sample which is mounted on the cryo stage, and thereby we can capture the growth of ice crystals and then draw it in pure water or CPA. Another powerful tool is a differential scanning calorimeter, where a small volume of sample can give us a thermogram which can be further used for calorimetric analysis, such as ice fraction, specific heat, and critical cooling and warming rates.
Alright, so my name is Casey. So here I'm just showing the iron oxide nanoparticles getting loaded into the heart. Up through here, through the aortic cannula, and exiting through the catheter and pulmonary artery. This is Z. After this step is complete, about 4 mils into the heart, she'll put it inside this bag here, which will then be placed inside the control rate freezer. And after that gets vitrified, we'll reform it by placing it inside this nanowarming coil.
We're currently using silica-coated iron oxide nanoparticles for RF nanowarming projects. We're able to scale up the synthesis from 150 ml to 4 liters. The silica-coated iron oxide nanoparticles and cryoprotective agents were loaded into red hearts and then vitrified in the control rate freezer and rewarmed in the RF coil. We're able to perform normothermic perfusion and record ECG for beating heart.
For organ vitrification and nanowarming, we perfuse IOMP into the organ and used micro-CT to detect the distribution. We also used the micro-CT to analyze the cross-section inside the organ and showed that there is no ice formation in the organ. We also developed metaform warming, which can be applied to tissue samples, for example, artery and skin, both of which showed greater viability compared to the conventional warming.
So a quick overview about the cryopreservation of tissue slices would involve harvesting the organ and slicing them into small 5 or 8 millimeter tissue cores. We can either use the steady rigs for rapid slicing, which gives us thicknesses of 500 microns, or a vibratome, which gives us thinner slices, 50 to 500 microns in thickness. Now the tissue slices are then diffused with CPA, vitrified, rewarmed, and the CPA is removed and the tissue slices are cultured. And now these slices are ready to be used for various applications, the most prominent ones being pharmacological studies, where these slices can be used for drug discovery.
Hi, I'm going to talk about what we do in this group, which centers around laser nanowarming. The main concept to understand is that we cool droplets with biological material in them down to cryogenic temperatures, and with the help of cryoprotectants, we can turn these materials into a glass, which avoids ice crystallization. We can then store them for indefinite periods of time. So we're working to develop protocols for banking of aquatic organisms such as coral larvae, shrimp larvae, and zebrafish embryos for conservation, aquaculture, and biomedical research. We're also working on human somatic and stem cells. Then, once these are stored and vitrified, we can use a high-power laser and 2 nanoparticles so we can rapidly rewarm the cryopreserved droplets to room temperature at millions of degrees per minute, outpacing ice on the way back and maintaining viability. Some systems are impervious to gold nanoparticles and require microinjection, as seen in this image on the left, or a golden shiner embryos being microinjected with gold nanorods and cryoprotectant. So in these cases, when irradiated by the laser, the warming is intracellular. In this next video, you can see Pacific white shrimp being vitrified and rewarmed with the laser. For cases like this, the gold stays outside of the cells and thus the sample warms extracellularly. This leads to lower warming rates, but is easier than microinjection, so it has its benefits as well. It's also important to study the toxic and osmotic effects that these different cryoprotectants have on different biological systems, such as zebrafish and Pacific white shrimp. To characterize how the system will warm under laser irradiation, one powerful tool that we use is Monte Carlo simulation. This allows us to simulate the photons emitted by the laser as they travel through the system, interacting with the boundaries, scattering, and getting absorbed by the nanoparticles, and as well as the material in the droplet. This allows us to calculate temperature profiles within the droplet, which can then be used for better experimental design. We've been able to show high agreeability with the model and actual laser warming. Notice the profile of warming and the location of hotspots in this high-speed video of droplet warming. The laser placement and droplet shape are also a few parameters that can be considered when we're optimizing laser warming. So you can notice there's a hotspot at the bottom that forms there, as well as the shell around. This image on the right shows the time evolution of that system compared to the Monte Carlo absorption profile calculated from simulations. We can also study the behavior of different nanoparticles under laser irradiation, with some scattering light more and some absorbing more. By using the previous Monte Carlo modeling and nanoparticle characterization techniques, we can optimize the nanoparticles so that we get the most ideal warming pattern possible. Then we can fine-tune our laser settings and attain ideal warming of vitrified droplets. As you can see, these are different nanoparticles here going across from A to B to C to D, and in C we have this optimal nanoparticle concentration, as well as photothermal properties. So the video for ideal warming, you'll see, shows a droplet being warmed at 3 million degrees per minute. So this is the ideal case where all the settings are fine-tuned. The second droplet shows what happens when the laser settings or gold concentrations are too large, leading to overwarming, possibly damaging or destroying the biological material within the droplet.
So one research project going on in the lab is to cryopreserve Drosophila embryos. So Drosophila, commonly known as fruit flies, is a model organism for different disciplines. Has been researched for over a century, the downside is that you need to keep that alive for adoption. So in the near future, we hope to cryopreserve them. This is the protocol overview of our procedures, and this is the outcome of our protocol.
Hi everyone, this is Pekka. I'm a PhD student in John Bishop's lab, and I'm working on characterizing miniature probes for preclinical cancer focal therapies. So what I'm essentially doing is, first of all, designing some focal therapy probes, which are small and meant to be used on small animal tumors. They consist of a cryoprobe, which freezes the tissue, a heating probe, which heats the tissue, and an IRE probe, which destroys the tissue by some electrical pulses. And then I go ahead and characterize them in porcine liver samples and also in mice tumor. And I do the characterization both experimentally and numerically. And what I'm essentially doing is is visualizing the energy field distribution around my probes, which in case of the thermal probes, the cryoprobe and the heating probe, it will be the temperature field distribution. And in case of the IRE probe, it will be the electrical field distribution. And then I go ahead and look at the verification of my experimental measurements versus my numerical model and see how well I've been modeling or measuring the temperature or the electric field around my probes. Thank you.
Hi everyone, I will introduce the rapid diagnostic project in Bishop Lab. Probably the most familiar application is pregnancy test. And our lab will synthesize those test strips on our wall, and we synthesize those colloidal gold in our white chemistry as labels, and develop those strips on our wall for improved assay performance. And hopefully, with this assay improvement and signal amplification by thermal contrast amplification, we would like to achieve future point-of-care tests that is both cheap and rapid and high sensitivity.
Hi everyone, I'm Jesse, and I'm also part of the Rapid Diagnostics project. Here you can see what we're doing is signal amplification of the lateral flow assays via what we call thermal contrast amplification. Basically what we do is we use a laser to amplify thermal signal and then record it via an IR camera. This lets us record signals subvisually.